Hello everyone, Mrs. Kleins here. Let's get started with learning about pH and enzymes in our next section of video notes. First, let's review what a chemical reaction is. A chemical reaction is a process that changes or transforms one set of chemicals into another. There are two parts in a chemical reaction, the reactants and the products. The reactants are elements or compounds that enter into a chemical reaction. They are seen on the left side of a chemical equation. The products are elements or compounds produced by a chemical reaction. They are seen on the right side of a chemical equation. During a chemical reaction, energy may either be released or absorbed during the chemical transformation. An exothermic reaction releases energy, usually in the form of heat. An endothermic reaction absorbs or gains energy and will not occur without a source of energy. Activation energy is the energy needed to begin a chemical reaction. In some cases, we need to apply heat to kickstart the reaction. The activation energy is shown here in an endothermic reaction on the left, and here in an exothermic reaction on the right. A pH scale is the measurement of the amount of hydrogen ions in a solution, which is why pH stands for potential hydrogen. The pH scale helps us to determine how acidic or basic a substance is based on the number of hydrogen ions. Therefore, a solution that is acidic would have more hydrogen ions, and a solution that is basic would have less. The scale runs from 0 to 14, where acids are represented from a pH of 0 to 6, and bases are represented from a pH of 8 to 14. Anything at 7 is said to be neutral. For example, pure water has a neutral pH. An acid is a solution that has an excess of hydrogen ions. It comes from the Latin word acidus, which means sharp or sour. The more hydrogen ions, the more acidic the solution. You have probably tasted an acid in your lifetime and not realized that acids have a characteristic taste. All acids, in fact, taste sour. They also can conduct electricity and are corrosive. Saying that acids are corrosive means that they can break down certain substances such as fabric, skin, paper, and so on. For example, some people will apply the soft drink Coke to help clean the car battery terminals. This is because Coke is an acid and it will eat away at the corrosion on the terminals of the car battery without, in fact, messing with the battery acid. Cleaning of the terminals on the car battery allows your car to start. There are many examples of acids you can probably think of, some of which are even helpful in digestion. However, here are some that you regularly interact with. Vinegar is an acidic substance. It is actually made up of acetic acid. Citric acid, which is found in lemons, limes, oranges, and even sour candies. Sulfuric acid is important in fertilizers, paint, and even plastics. Lastly, car batteries are composed of sulfuric acid, which helps to provide an electrical current to start your car and also recharge the electrical system while the car is running. A base is a solution that has an excess of hydroxide ions. Bases are also referred to as alkaline, so oftentimes another word for a base is alkali. Bases are always substances that can accept hydrogen ions. You may have never realized it, but bases, just like acids, can have specific characteristics. Bases always have a slippery texture and will have a bitter taste. They are and can become as equally corrosive as acids, especially depending on how strong the base actually is. Interestingly enough, bases can also conduct electricity. Like your Energizer batteries are known as alkaline batteries, meaning base batteries. Bases, however, do not react with metals like some acids do. Examples of bases include household items such as soaps and ammonia. Chalk and oven cleaner are also examples of products that contain bases. Even milk and blood are slightly basic. A change of 1 pH unit represents a tenfold change in the acidity of the solution. For example, if a solution has a pH of 1 and a second solution has a pH of 2, the first solution is not twice as acidic as the second, but actually 10 times more acidic. Another important feature of pH scale to consider is that acids and bases can neutralize one another. A reaction between an acid and a base is called a neutralization reaction. because the mixture is not as acidic or basic as the individual start solutions. For example, in the picture in the upper right-hand corner of your screen, the base has a pH of 12 and the acid had a pH of 2. When the two are mixed together, a more neutral solution with a pH of 8 is produced. The pH of a solution has a great impact on the effectiveness of the chemical reactions that take place in them. For example, your stomach is composed of gastric acid that has a pH from 1.5 to 3.5. All of the chemical reactions that take place in your stomach have to be able to withstand this very low pH. If the pH suddenly became more basic, then the chemical reactions would stop and you would become sick. Enzymes are one of those substances that require a specific pH in order to function correctly. Enzymes are an example of a protein that resides within cells, and for the most part, they all end in ASE or ACE, which makes them easy to identify. Some examples of enzymes are sucrase, catalase, lactase, amylase, and of course, there's always an exception, pepsin. Enzymes are extremely efficient at catalyzing or speeding up chemical reactions. This is their sole job within a cell, to break apart and put together various compounds via their chemical reactions. And they do this by lowering the activation energy. This is the energy that is required to start a chemical reaction. Therefore, there's less energy being expelled by the cell, which is great because then the cells are not wasting energy. This graph shows the activation energy needed for a reaction to occur without an enzyme, with the red line, is more than the activation energy needed with an enzyme, shown by the black line. If you recall, what goes into a chemical reaction are the reactants, these are on the left side, and what comes out of the chemical reaction are the products, these are on the right side, as seen in the equation in the upper right-hand corner. An enzyme works by the reactant known as a substrate attaching to the enzyme at a particular location known as the active site. This is where a chemical reaction takes place. The products get released from the active site after the chemical reaction is completed. Now, if molecules are going to be joined together, then the reaction is known as dehydration synthesis. This is where multiple substrates will form one product and one water molecule will be released for every bond that is formed. On the other hand, if the substrate is going to be broken down, like in the animation on your screen, then a hydrolysis reaction will take place that requires one water molecule for every bond that needs to be broken. In the case of the animation on your screen, only one water molecule is necessary because only one bond is broken. Once completed, the products are released and the enzyme will wait for the next substrate to complete the next chemical reaction. Now it is important to note that the shape of the active site on an enzyme only fits the shape of one specific substrate or reactant, similar to how a lock fits a particular key. Only the correct substrate binds to the correct enzyme. Therefore, there are thousands of enzymes that must exist for all of the substrates for chemical reactions that take place in our cells. Enzymes themselves can be manipulated too, intentionally or inadvertently. For example, if you want to speed up a chemical reaction that is taking place, then you can add more of the enzyme, and the enzyme will continue to process the substrate until there is no more substrate left. You can slightly warm the enzyme as well to speed up the chemical reactions. However, you do not want to do that too much. Just like pH can mess with an enzyme, temperature can too. All enzymes have an optimum or best pH and the temperature that they function at. You can see this represented on your screen here, showing the optimal temperature for two different enzymes. A change in the pH and/or temperature can cause the active site of an enzyme to denature or change shape. This will then prevent the substrate from binding to the enzyme's active site. The substrate will accumulate in the organism's cells until the shape of the enzyme returns to the normal shape, or the organism may get sick and die. There are also several genetic disorders that are caused by DNA mutations coding for the incorrect shape of particular enzymes. This also prevents the appropriate chemical reactions from taking place in those cells, and the substrate accumulates in the body of the organism as well, usually leading to a tragic outcome. All right, folks, so we are done with this section of your video notes. Be sure to ask questions if you have them and take the time to review your notes.